talking about the life cycle of hepatitis B virus. So hepatitis B, which is a very, very dangerous virus, which is causing a disease which is kind of being carried by 1/3 population of the whole world. So you can see how dangerous this hepatitis B virus can be. So hepatitis B actually causes the hepatitis disease, which is related to hepatocytes or the liver cells. It can cause liver failure and liver cirrhosis and many other liver damages and problems of your body. So normally hepatitis B virus is known to be a very, very small virus, very, very tiny virus particle with obviously a DNA. And the DNA that this hepatitis B is carrying, it's a double-stranded DNA. And this virus is again, though it's a DNA-carrying virus, but it is having a slightly different mode of life cycle and transmission inside the host cell. And it is required an important enzyme for its replication inside the host cell, and that is called the reverse transcriptase. That's why this virus is also called DNA virus, or we can also call it the retro type of virus. Okay, so it's a kind of retro type of viruses because it requires to synthesize DNA from RNA templates. So it's kind of retro type, and it is called the HEPA-DNA virus. So HEPA DNA virus. Okay. So the major component of this virus, if I list the major components are obviously this virus is very tiny. So it's having a capsid. So inside this virus capsid, so I'm not drawing capsid in different section, I'm drawing it circular. So this is the capsid of the virus, and inside the capsid of the virus, it is having its genetic material, which is tightly coiled like that. And at the end of the genetic material, it is also having attached with polymerase. And this is the RNA polymerase that is required by this hepatitis B virus for the basic induction and production of important proteins for its replication inside the host cell. So it carries its own polymerase enzyme with it. So if this is the content, so let me mark them. This is the polymerase enzyme, and this is nothing but RNA polymerase, and this is the double-stranded DNA, which is also small, of the hepatitis B virus. And it is having kind of circular type of viruses, which is super coiled like that. So it's a kind of circular. Okay. So it's kind of covalently closed circular type of DNA. So CCC DNA is there. So this virus, when it infects, normally it comes into the contact and it normally spread by the blood transfusion. And normally when it is traveling through the blood, it will reach the hepatocyte or the liver cells, and it will infect the liver cells. So when it is inside the liver cells, then it will start to do the molecular trickeries that we are going to see now. But remind you, the process of entering inside the liver hepatocyte cells is not totally clear. So I'm not going to talk about that. So this virus usually enters through this through the cell membrane of liver cells. Maybe receptor-mediated endocytosis is there. So after engulfed inside the liver cells, this virus starts to play the role. So let me draw this. So if this is the liver cell surface, and here comes the virus, and the virus, say this virus enters inside this liver cell cytoplasm. So here comes the virus, and inside the virus particle, we know we are having the DNA materials here like that, and at the end of the DNA we are having also the RNA polymerase. Then it will move through the cytoplasm using the microtubule. We all know the importance of microtubule, right? So microtubules are required to take them there. So we require microtubules. So using microtubules, they will migrate towards the nucleus. Remind you, the nucleus is placed here. So here comes the nucleus. It is the nuclear membrane. So it will migrate towards the nucleus. And once it is migrating towards the nucleus, this outer coat, it is called the core section of this virus particle. The core section, it starts to dissolve. So as the core section starts to dissolve, so let me write core dissolves, and then it will release its particles inside this nucleus. What are those particles? Two important things, remember. One is this tightly coiled DNA and this polymerase. So these two things are now released inside the nucleus. Okay. Now the host cell nucleus is also having the host cell genome, but I'm not writing that, I'm not drawing that because for your understanding sake. So here we come, these two things. Now this particular supercoiled DNA of this virus is now relaxed a little bit. And what it forms, it actually starts to coil with each other. Because normally at the very beginning, from this beginning, actually I'm not talk completely correct, because these are not completely supercoiled. These are supercoiled, but excuse me. These are supercoiled, but these are not covalently linked at this point when they are present in the virus or virion particles. Now once they are inside the nucleus, then only they are coiled. Okay, they are attached. The terminal portion of this single-stranded, the terminal portion of the double-stranded DNA is now attached. So the DNA is linear, but they are supercoiled. But now the terminal portion will be attached with each other. So the terminal portions will be attached here using certain protein molecules. So certain enzymes will join them. So now we get a covalently closed circular DNA inside the nucleus, and then we have the virus RNA polymerase. Now this viral RNA polymerase, what it will do, RNA polymerase of virus, it will sit there, and what it will produce viral mRNA. Many viral mRNA will be produced there, right? So once they produce this viral mRNA will be brought inside the cytoplasm, because remember, translation always occurs in the cytoplasm. So mRNA will be here. From the mRNA, they start to produce many important protein factors. One of those important protein factors is the DNA polymerase of this particular virus. The DNA polymerase of hepatitis B remind you, is not a commercial and common type of DNA polymerase. It's a special one because this DNA polymerase is having three activities. One is the activity of priming things, because we know normally DNA polymerase cannot prime, cannot initiate the process, but here they can prime, they can do a reverse transcription, and also they can complete the whole process by sealing the nick. So this is a very, very special DNA polymerase. So you get this DNA polymerase produced from here, and then also they produce certain type of proteins that are required to produce the structural length, right? So structural proteins will also produce. So once the structural proteins will be produced, they will be taken inside the nucleus. Remind me, they will be taken inside the nucleus. But now all these DNA polymerase things and everything, they will also be taken inside the nucleus. So those proteins are then taken inside the nucleus. All of these proteins. So all of these proteins will be taken inside the nucleus. Now once inside the nucleus, let me stretch the nucleus a little bit. Okay. So once inside the nucleus, this DNA polymerase will use this mRNA. This mRNA, remind you, the mRNA that they produce at the very first place are called pre-genomic mRNA. This is called pre-genomic mRNA. So once they produce this pre-genomic mRNA, then they produce DNA poly with it, and then everything is inside now. Now this DNA poly that is inside now, this poly will start to produce a DNA, a double-stranded DNA, using this pre-genomic mRNA. And here comes the importance of reverse transcription, because we know normally transcription means DNA to RNA, but when it is RNA to DNA, it definitely is reverse of the transcription. So they will take this mRNA, pre-genomic mRNA, they will start to produce a DNA with it. So what they will take here, they will take this mRNA. So this mRNA will be taken as a template. Remind you, this mRNA which is present here, this mRNA is having a special region, and this is called a stem loop region, like that. So using the stem loop region, this particular polymerase, so let me draw the polymerase in higher amounts. So the polymerase will look something like big. It will look big. So here comes, so let me erase this part and draw it bigger. This is the polymerase. Now once this polymerase detects this stem loop structure and its stem loop of the RNA, now what it will do, it will detect this RNA stem loop and it will replace a simple codon there. And here at this point of the junction, there is a priming site of this polymerase. Remind you, I told you that this RNA polymerase have three functions, three major functions. This DNA polymerase, sorry. One function is the priming of this. Second function is the reverse transcription of it. And third function is completing the dsDNA synthesis. So the first place is done by the priming section, where it detects the stem loop and it attaches a guanine there. First of all, it attaches a guanine, then it attaches two adenine side by side. Now this particular point of guanine and two adenine acts as a primer for the synthesis of first DNA strand. So then the first DNA strands will be synthesized by taking this genomic RNA or pre-genomic RNA as a template via the reverse transcription process, remind you. So let me draw it, what is going on here. So we are having simply we are having the RNA here. And we just done the priming here. After draw the priming, we start to produce a first strand of DNA. So let me draw DNA in this way. So first strand of DNA is synthesized now, like that. So once we're synthesizing the strand of DNA using this as a template, but at the end of it, remind you, this is an RNA-DNA hybrid now. Now what is going on, during the synthesis of DNA, they are also simultaneously cutting the RNA strands because they don't require the RNA strand anymore. But, so after a certain amount of time, what we'll be having a DNA here like that. So we are continuously producing this DNA simultaneously. So obviously, let me put this primer here. And along with that, we are degrading this RNA because we don't require that anymore. So ultimately, we completely start degrading this RNA. But at the end of the RNA, we maintain a section of RNA, because this ultimate end of the RNA, which is having a free hydroxyl at 3', will serve as primer for synthesizing the next strand, right? So that's why the virus takes this last part of the RNA intact, right? So again, it's an RNA-DNA hybrid, but simple section is kept at the end. Then after we have this process, then what we get, so let me draw it again. So we are having simply this is a section of RNA, and in this point we are having this DNA attached, this primer attached, and the DNA, remind you. So this is the structure. And now we take this virus, take this primer, this RNA as a primer, and then it will start synthesizing the other strand of the DNA. And this is the positive strand, remind you. This other strand of the DNA will be synthesized now. So once it is synthesized, it will be circularized. So this DNA, once both the DNA strands are synthesized, positive and negative, so now it is becoming double-stranded DNA. Then it will be circularized, right? This DNA will be made to form a circle like that. Okay. DNA circle will be made. So now it is CCC DNA again. And then this DNA is ready to be packaged inside the phage particles. And I remind you, inside the virus particle. And I remind you that during the process of this production of from the genomic RNA inside the cytoplasm, we not only produce DNA polymerase, but also we produce some structural proteins which are also taken inside the nucleus. So here, the structural proteins start to emerge. They start to coat this DNA. So now there will be a packaging. The structural proteins start to package everything up, and they will package things. Inside it, we'll be having our closed, circular, and also supercoiled DNA. At the end of it, the terminal part, we are having the polymerase, and everything is packaged. And a progeny virus is now ready to emerge. Now, during this emerging, this virus is also moving through endoplasmic reticulum, then through Golgi apparatus. Finally, they are released outside. Remind you, this packaging takes place in the nucleus, then they migrate towards ER, then they migrate towards Golgi, then finally they just break open the cell membrane and come out, right? So this is the overall process of how this whole process of hepatitis B virus is replicated, and how hepatitis B is actually causing the infection in us, and that's kind of it. And I hope it's helpful. Thank you, guys.
The ACE Innovation $15 home STD screening kit detects the hepatitis B virus.